That way I can just tell him to watch this later if he doesn't get back in here.
Would it work if one of us called, like they send in or called in and then I set him up here for me and you could see the
Maybe. Let's see if he pops back in. I think he may pop back in. All right, let me go through this. This is the support. And you may not be able to see that from where you are. Don't worry, I'll dictate this. So the video mentioned these things. These are supports for endosymbiotic theory. Here we go. Mitochondria and chloroplasts, right? That's what we're dealing with. Mitochondria and chloroplasts. The support is, or the interesting thing is that they have their own DNA. That is true. If you look internally, inside the structure of a mitochondria, it has its own DNA. It's called mitochondrial DNA. Chloroplasts have their own DNA. So that's kind of a curiosity. When you look inside of these structures, mitochondria and chloroplasts, they actually have their own DNA that is separate from the DNA inside the nucleus. Number two, and you see I don't want to have to write that again. So number two, mitochondria and chloroplasts have different membranes. They have several membranes. That is true. And mitochondria has what's called an outer membrane and then an inner membrane that has what's called cristae. You don't have to remember all this now, but the cristae are the little folds of the inner membrane. In other words, there are several membranes of a mitochondria and also of a chloroplast. That is true. Number three, mitochondria and chloroplasts have their own ribosomes. That is also true. You look internally inside of a ribosome or of a chloroplast or mitochondria, you will see that they have ribosomes. Now, what do ribosomes produce? Proteins. Now, that's important because if this used to be a free-living organism, it would have to make its own proteins, right? So if it was incorporated and made into an organelle, the fact that we find ribosomes might make sense of that, right? Okay. And then lastly, chloroplasts and mitochondria can reproduce on their own. In other words, they do not have to wait for the entire cell to replicate. They can actually make their own copies. In other words, mitochondria can go kind of enter their own replication process and make copies of themselves internally inside the cell. So these are the major supports. If you say, what are the supports for endosymbiotic theory? These are the major ones right here. Okay? Now, we're going to come back to that, okay? Or we'll touch on these again. I just want to kind of leave it there at the moment because I want you to read this article that I put in front of you. There's actually two articles there. You don't have to read both. You can read one or the other. The first article says Endosymbiosis, a theory in crisis, and the second one, a study demonstrates complex cells could not evolve from bacteria. Now, these are your articles. You keep these, so you can write on these, you can underline, you can highlight, do whatever you want. What I would like you to do is, as you read the article, just maybe jot down a few of the difficulties that they mention. Okay? So we just went through some of the support. Now let's get some of the difficulties. And then on the other side of this, we'll kind of put all this together, right? Here's some support for it, here's some difficulties, and we'll think through some of the questions, because I guarantee you, new questions will come to your head and mind, and so we'll walk through those as well, okay? So let me give you just a little bit of time here to read through one or both of these articles. If you just read one, just read the first one, I guess, the top one. But if you want to read the second one, that's great. Or later, if you want to read the second one, that would be good too. So anyway, let me be quiet for a few minutes here and let you just read through that. Okay, so let's think through some of these difficulties. We've talked about some of the support. So what are a few things the article mentioned that might be difficult for this endosymbiotic theory? Grace.
It definitely makes sense how it could just like randomly switch once the time stops.
Okay, good. The genes that are retailored, it mentioned that. And a lot of this was talking about certain genes and how the genes between, you know, if there's one cell that's larger that has to engulf the smaller cell, well, okay, then there's going to have to be some sort of alignment or coordination between the genes of the larger cell and the smaller cell that was engulfed, right? They have to, somehow that's all going to have to coordinate. Good. Go ahead, Maddox. Yeah.
You also said that there is no display of steady evolutionary progression for simple life.
Okay, good. Steady evolution. That's a question. In other words, actually, this is quite interesting if you think of it. Normal evolutionary process would state that if this were what they were considering a simple cell to a complex cell, normal evolutionary process would say, well, that simple cell slowly, through mutations over a lot of time, became a complex cell, right? Slowly over time, that's what happened. That simple cell became a complex cell. But that's not what endosymbiotic theory is suggesting. What is endosymbiotic theory suggesting? Is it suggesting that slow change over time? It's actually suggesting a sudden change, right? What's the sudden change? Yeah. Here's a larger cell that engulfed a smaller cell. In other words, this larger cell didn't have those organelles. And then it engulfed a smaller one, which became the organelles. In other words, it's trying to speed up the process of explaining how the organelles came to be. Does that make sense? Instead of trying to explain how mitochondria and chloroplasts evolved on their own slowly over time through mutation, it's not trying to explain that process. It's trying to explain how mitochondria and chloroplasts appeared suddenly. Does that make sense? Okay. Any other complications? These are all good. Yes. Grace.
So is it that they're trying to explain something that's not so
This would still be an attempt to explain how mitochondria and chloroplasts came about through an evolutionary process. But actually, it's not pure naturalistic evolution. The mechanism isn't, because it's not by slow development. Say slow development would be, okay, that simple cell kind of developed a structure kind of like a mitochondria, and then over time it was more like a mitochondria, and then eventually it became a mitochondria. That would be kind of a normal slow evolutionary process. This is actually speeding it up and saying it wasn't slow. This larger cell actually engulfed a smaller cell, and that smaller cell became the organelle. Does that make sense?
Is the problem with that it just happens to not make sense?
Well, of course, evolutionists would say there's no problem with that. I think that the objection would simply be that is now circumventing normal evolutionary processes. Does that make sense? Yeah, circumventing meaning it's kind of going around the normal process. Does that make sense?
Yeah.
The normal process is slow, steady change, and this explanation is like circumventing that and going around normal slow change, and trying to explain how evolution happened all at once. Yes.
Well, I mean, I don't think it's really circumventing evolution because it's still two things that have evolved all around and then they just combined. So I don't think that it's like fairly irrational thing. Like,
Well, I understand what you're saying. I totally understand. And they of course, naturalistic evolutionists would say that's fine. Whether they would say circumventing or not, but what they would agree to is that endosymbiosis is not an attempt to describe a normal mutational type of evolution. In other words, they're not trying to explain how mutations produced a mitochondria, or they're not trying to explain how mutations produced a chloroplast. They're not trying to do that. They're trying to explain how mitochondria and chloroplasts appeared in a eukaryotic cell, not by slow processes, but kind of by all of a sudden by engulfing it. Now, they would still say, okay, we still think it happened.
yeah, well, if that's really what happened, then they shouldn't be trying to mold their, because, I mean, yes, they could say that they just developed, but that's not really what their evidence suggests happened, then why would they say that? It doesn't make sense to try and mold the evidence to your conclusion, you know?
Yeah, I think that maybe the objection is simply, so as an objector to that, I would look at the person who's suggesting endosymbiotic theory, and I would say, I understand what you're saying. In fact, actually, what you're saying makes some sense to me. But then the objection would be, but what you're telling me is not following the normal evolutionary process that you normally suggest. You're circumventing. Does that make sense? Yeah. Okay, any other thoughts? Okay. Maybe, do you have any other general objections or difficulties? Maybe these are not even found in the article, but you're just kind of thinking through the process now, and some things are coming to mind that might be difficulties or objections. Any of those coming to mind? Yes.
So, I'm not sure if this was covered or anything, but when a cell replicates itself, right,
Yes.
it transfers everything that it has with it, right? It doesn't create, maybe the membrane and then mitochondria replicate itself.
Correct.
So yeah, that was one fan of it. It does say that in the theory that it becomes an organelle, but how exactly will that happen? And if they were to explain it, you could then find a way to say, okay, that makes sense.
Right, right. So I think you were out just a moment. So Grace mentioned something about the coordination aspect of it. Genes retailored or the coordinate. In other words, it couldn't be simply that a larger cell engulfed a smaller one and then everything was just happy-go-lucky at that point. Because if that did happen, then there would have to be a lot of coordination between the smaller cell that was consumed and the larger cell that consumed it. Now there's got to be a lot of coordination that takes place. And that's a little tricky to explain how all of the coordination would have taken place. Is that what you're getting at?
Yeah.
Yeah. Okay.
So another difficulty that I found in the article is that essentially all the first eukaryotic cells would have had to have a fully functioning mitochondria.
Oh yeah. Okay. So you say then we're at kind of back at the same level where, yeah, that's a good point. Fully functional mitochondria or chloroplasts, either one, right? In other words, it would do no good for that larger cell to have engulfed a proto-mitochondria, something that didn't function as well as it should have. Does that make sense? In other words, it only makes sense if that larger cell is consuming something that already works perfectly and can easily be incorporated within the broader cellular structure. Does that make sense? What I think, Maddox, is that what you're saying? Yeah. Right? That's a good point. It would have to already be a fully functional organism, which became a fully functional organelle, right? Okay. Any other thoughts, General? Yes.
Also, it said that for it to do all the retailoring and everything, it would have to have a significant amount of energy that bacteria could not acquire.
Yeah, the energy, because if you think, well, if that living organism that became the mitochondria was functioning on its own, it makes sense that it could produce the energy for itself, right? In other words, that little smaller organism produces a certain amount of energy that controls its cellular function. But now it's inside of a larger cell, and that larger cell is going to require a lot more energy. So how did it produce enough energy for the larger cell? I think that's what you're talking about, yes. Yeah.
The article also said that the first eukaryote has this mitochondrial membrane of sufficient surface area. All known mitochondria have very convoluted and folded membranes to keep this unlike the bacterial membrane, which is supposed to be evolved.
Yeah, so that's good. Mitochondria are kind of like this. Mitochondria have several membranes, but it looks more like that. Did I say mitochondria? I meant bacteria. Bacteria have two membranes and look kind of like this. This is a bacterial cell. Mitochondria have two membranes, but it looks more like this. In other words, that inner membrane is folded all over. It's what we call cristae, but it has a lot of folds. In other words, the membranes between the two are not 100% identical. So in other words, why did the membrane shift or change? That's a question. Good. Any other thoughts?
What would number 5 be?
I just put a certain amount of numbers up here. I didn't know how many we would come up with, but I mean, I can throw a few more in here, but I didn't know how many you guys would say. Here, I want to go to here just a second. The reproduction. It is true that mitochondria and chloroplasts can reproduce on their own. Can you think of a benefit for a eukaryotic cell to have mitochondria or chloroplasts that can reproduce on their own without having to wait for the cell to reproduce, the whole cell? Grace?
Well, if they produce on their own, it's probably produce more, so there'll be more organelles that produce.
Good, good. So, in other words, what I guess what you could say is this last point, the reproduction on their own of mitochondria and chloroplasts, that is not an exclusive benefit just for the evolutionary process. Does that make sense? In other words, we would explain that for any eukaryotic cell that would be a benefit. In other words, if I had a muscle cell and that muscle cell needed a lot more energy, could the mitochondria in my muscle cell produce more mitochondria that would produce more energy without the muscle cell actually replicating? The answer is yes, that can happen. In other words, actually muscle fibers are really long fibers. They're really big cells. So what happens is the mitochondria can replicate without the cell having to replicate. You see how that could be a benefit? So all I'm saying there is actually that point makes sense either way you go, right? It makes sense if endosymbiosis is true, but it also makes sense in normal eukaryotic cell function. That makes sense. So that's not like an exclusive description of the endosymbiotic theory. Can you think of a reason why DNA or ribosomes might also be a benefit for any eukaryotic cell normally? Ribosomes, again, produce what? Proteins. Are there a lot of proteins necessary to produce energy for the cell? What do you think? Yes, there are a lot of proteins that are necessary to produce energy for the cell. So the fact that mitochondria can produce their own proteins related ultimately to the energy that they have to produce makes perfect sense. And actually what's controlling the production of proteins is the instructions in the DNA. So the fact that mitochondria have their own instructions that help produce their own proteins so that they can produce energy, all of that's kind of a package deal. Does that make sense? You kind of can put all that together as a package deal. So again, all of these major supports for endosymbiotic theory might in fact also be explained quite easily through a creation mechanism, right? In other words, God gave mitochondria and chloroplasts this ability so that they could perform their task to the level that they need to perform their task. Okay, any other last thoughts here? We're doing good. These are really good thoughts, really good points. I'm going to add a number 5. I think some of you thought of this, you just haven't said it yet. Endosymbiotic theory explains what two organisms came about. Chloroplasts and mitochondria. Yes, but are those the only organelles that eukaryotic cells have? What else do they have? Nucleus, membrane, endoplasmic reticulum, Golgi body, lysosome, right? And we're still leaving out a few. In other words, maybe endosymbiotic theory can explain how chloroplast and mitochondria came to be organelles. It doesn't do anything to describe how endoplasmic reticulum got there, or Golgi bodies got there, or anything else, right? Does that make sense? In other words, though endosymbiotic theory might explain part of the formation of a eukaryotic cell, it can't explain the whole formation of a eukaryotic cell, because it certainly doesn't explain how those other organelles came to be. So at best, it can only explain part of the scenario, part of the puzzle. It can't explain the whole thing. At the end of the day, you still got to say, well, where did the endoplasmic reticulum come from? Where did the Golgi bodies come from? Where do the lysosomes come? Okay, does that make sense? Any other thoughts here? Now, saying all of this, I lay it out in front of you because it's a very interesting, I would admit, it's a very interesting theory. It's a very interesting hypothesis. You know, we're dealing with living structures, larger cell that can consume a smaller cell. Is that possible? Well, we do see larger cells consuming smaller cells, but typically that's for nutrition, right? So maybe one other question is simply this. If this happened in the past, why are we not observing it happening when? In the present. In other words, if it happened in the past, why would it just be something that just happened in the past and not still happening in the present? Why don't we observe this sort of thing happening often today? Yes. Or at least sometimes today, yes.
Because maybe the cells are fully complete. There's something else that they can do in more space.
Yeah, maybe they're already complete. So I mean, there's no need to take in anything else. Okay, yes. Yeah. But that is you say scientifically that's a valid question. If it happened then, was it just an isolated event in the past? Why is that not something that we can observe today? Maybe I'll give you an example of that. We have bacteria inside of our intestines. I think you know this, right? What do the bacteria inside of our intestines do? What do they help us with? Digestion. That's a symbiotic relationship, right? In other words, there's a symbiotic relationship between our cells in our intestine and the bacteria that are inside of our intestinal tract. There's a symbiotic relationship. But what we don't observe happening is our intestinal cells actually engulfing the bacteria and doing what? And keeping those bacteria for their own purposes. Does that make sense? In other words, the bacteria in our intestines are still bacteria in our intestines. Our intestinal cells have not engulfed them and now kept them for their own purposes. No, they're still separated, right? In other words, that would go into explaining why, you know, why don't we observe this sort of thing happening today? Any last thoughts? Okay, all right. That was good discussion. So see,